Hello everyone. Today we are going to start a new topic, that is plasma membrane. In this we will discuss about lipid component of plasma membrane. We will discuss about some of the important characteristics of plasma membrane. We will discuss about the protein component of plasma membrane. So let us start. Plasma membrane, it is outermost boundary of cell. Cell is the smallest unit of life. It is structural, functional and biological unit. Plasma membrane is selectively permeable. That means it does not allow the movement of all the molecules. Only selective solutes and solvents are allowed to move. And in this way it controls the molecular trafficking and thus maintains the internal composition of cell. There are various models proposed by different scientists to explain the structure of the plasma membrane. Among them the most accepted is the fluid mosaic model proposed by Singh and Nicholson. It is most accepted. According to this, proteins like mosaic are embedded in the lipid bilayer. This is lipid bilayer. In this, proteins are embedded like mosaic, and plasma membrane also shows mobility, thus it is fluid in nature. Dynamic fluid like structure. Plasma membrane shows mobility. Lipid shows more mobility compared to the protein because proteins are larger molecules and have more molecular weight. Chemical constituents. Plasma membrane is basically composed of lipids and proteins. In addition to lipids and proteins, it also contains carbohydrate in small amount, that is approximately 5 to 10%. This carbohydrate is present in the form of glycolipids and glycoproteins. When the carbohydrate is bound to lipids, it is known as glycolipids, and when it is bound to proteins, it is known as glycoproteins. Carbohydrate is present in abundance in the plasma membrane of eukaryotic cells. Now let us see about the lipid component of plasma membrane. There are three types of the lipids present in plasma membrane. One is phospholipid. This is the major lipid component. Second, glycolipid. And third is sterols. The first is phospholipid. These phospholipids are amphipathic molecule. That means these have polar head and non-polar tail. This is polar head and this is non-polar tail. Polar head is hydrophilic. That means it is water loving or soluble in water. And non-polar tail is hydrophobic. It is not soluble in water or water hating. This shows the kink in the fatty acid chain. That means it is unsaturated or double bond is present. Now, how the phospholipids are arranged in the plasma membrane. Phospholipids are arranged in the form of the bilayers. These are present in the form of the bilayers. This is one leaflet and this is another leaflet. Here the non-polar tail are facing each other and this forms the hydrophobic interior of plasma membrane, and the polar head faces the aqueous phase, and this aqueous phase may be external or internal. It means that suppose this is the cell, this is cytosol, and this is the outside region. So this polar head, it is facing the outside of the aqueous phase and it is towards the inside. This is also aqueous phase, and these tails form the hydrophobic interior of the plasma membrane. The force that hold these two phospholipid bilayer is the hydrophobic interaction between the non-polar tail. Phospholipids. It is major lipid present in plasma membrane. It is of two types: glycerophospholipid and second is sphingophospholipid. In the glycerophospholipid, it consists of glycerol, then two fatty acid chain is present, and phosphate and alcohol is present. This forms the polar head, and this fatty acid chain forms the non-polar tail. In the sphingophospholipid, sphingosine, is present one fatty acid, phosphate and alcohol is present. This is fatty acid, phosphate and alcohol. Here this forms the polar head, and this is non-polar tail. Glycerophospholipid. It is most abundant phospholipid in plasma membrane. Glycerol, two fatty acid chain and phosphate together known as phosphatidic acid. This phosphatidic acid plus alcohol forms glycerophospholipid. Depending upon the alcohol, the names are given. If the alcohol is serine, then it is known as phosphatidyl serine. If the alcoholic group attached is choline, then it is known as phosphatidylcholine. It is also known as lecithin. If it is ethanol amine, then it is known as phosphatidylethanol amine or cephalin. If the alcoholic group attached is inositol, then it is known as phosphatidylinositol. This phosphatidylserine is most abundant in plasma membrane. Lecithin and cephalin is abundant in plasma membrane of animals. This phosphatidylserine and phosphatidylinositol are negatively charged. This phosphatidylcholine and phosphatidylethanol amine has 0 charge. So the charge varies. The second phospholipid that is present in plasma membrane is sphingophospholipid. It has sphingosine instead of glycerol, and one fatty acid chain is attached to the C1 carbon, hydroxyl group is attached, and this is known as the ceramide. That is the precursor of sphingophospholipid. Ceramide plus phosphate group plus alcohol is known as sphingophospholipid. When this alcohol is choline, then this sphingophospholipid is known as the sphingomyelin. Second type of the lipid that is present in plasma membrane is known as the glycolipid, in which sugar residue is covalently attached. Glycolipid is again of two types. One is known as the sphingoglycolipid. Second is the glyceroglycolipid. This sphingoglycolipid is also known as the glycosphingolipid, and glyceroglycolipid as glycoglycerolipid. So the structure is sphingosine it has one fatty acid chain and carbohydrate is covalently attached. In glycerophospholipid, glycerol to fatty acid chain and carbohydrate. In the sphingoglycolipid, to the ceramide, that is the precursor, carbohydrate is attached. Sphingoglycolipid is the major glycolipid present in plasma membrane. It is then classified into cerebroside containing single sugar residue and globoside which contains 2 or more sugar residue. This cerebroside is classified into glucocerebroside and galactocerebroside. Glucocerebroside, if sugar residue that is attached is glucose, and galactocerebroside if galactose is attached. Then globoside. This globoside can contain sialic acid, that is with sialic acid, and it can be without sialic acid. If it is with sialic acid, then it is known as the ganglioside. This sialic acid is complex oligosaccharide. Known as N-acetyl muraminic acid. Third type of the lipid that is present in plasma membrane is sterol. It is amphipathic in nature. It is structural lipid. It is present in the plasma membrane of eukaryotes. It is rigid, planar molecule. It consists of 4 fused rings. Among them, the 3 rings are 6 carbon and the 1 is 5 carbon. This is known as the steroid nucleus. To the third carbon, first, second and third, hydroxyl group is attached. Derivatives of sterols that are present in plasma membrane. First is cholesterol. This is present in plasma membrane of animals. Second, ergosterol, plasma membrane of fungi. Stigma sterol, plasma membrane of plants. We are going to see some of the important characteristics of plasma membrane. Let us see about lipid rafts. We have seen that in plasma membrane, different kinds of lipids are present, but these are not randomly distributed in phospholipid bilayer. Some of the lipids like cholesterol and sphingolipids, mainly glycosphingolipids, are present at aggregate at certain places in plasma membrane. And these are known as lipid rafts. These are actually microdomains. These also contain specific proteins. Here we can see that this is phospholipid bilayer. At this region, cholesterol and glycosphingolipids are present. This is known as lipid raft. These are microdomains of plasma membrane. These are involved in signal transduction, endocytosis, and cholesterol trafficking. We will study these three in detail in separate topics. Let us see how the lipid molecules move in the phospholipid bilayer. It shows three types of movement or motion. First is rotational motion. In this, the lipid molecules rotate along the long axis. This is lipid molecule, this is its long axis, so it will rotate like this. This type of the motion takes place. Second is the lateral motion. This is lipid molecule and it moves laterally within the leaflet, and it is responsible for fluidity of membrane. These two motions occur within the leaflet of phospholipid. Third is the flip-flop motion. In this type of the motion, lipid molecules move between the bilayers. Suppose this is two leaflets of the lipid bilayer. This lipid molecule will move to this leaflet and this one will move to this leaflet. So they are changing their position. This is rare motion or rare event, because here the polar head has to move through the hydrophobic interior. Because it is rare event, so it maintains the asymmetry of phospholipid bilayer. Plasma membranes show asymmetry. It means phospholipids are asymmetrically distributed in the lipid bilayer. Amine containing phospholipid are present more on the cytoplasmic surface. Choline containing phospholipid and sphingolipids are present more on external surface. But the distribution of phospholipid is not same in all the plasma membrane. It may vary from the plasma membrane of cell to cell. We can see the example of erythrocyte plasma membrane. It shows the percentage of phospholipids. This is the total phospholipid. Sphingomyelin and phosphatidylcholine is present more on the exterior surface of the plasma membrane, and phosphatidylethanolamine and phosphatidylserine are present more on the cytoplasmic surface of plasma membrane. Plasma membrane also shows fluidity. This fluidity is required for the cell growth and normal functioning. There are various factors that affect the fluidity of plasma membrane. First is the temperature. As the temperature increases, the fluidity of the membrane increases, and decrease in the temperature causes the decrease in the fluidity, and these become more organized and gel-like. Change in the fluidity of membrane due to change in temperature is called phase transition. And temperature at which this transition takes place is known as the transition temperature. Second is fatty acid chain length. If the fatty acid chain length in the phospholipid is less, that means if the fatty acid chain length is short, then the surface area for interaction is less, and hence there will be lesser van der Waals force of interaction. So it will be more fluid because they are less tightly packed. But if the fatty acid chain length is more, so there is more surface area of interaction, hence more van der Waals force of interaction is present, and thus they are more tightly packed. Therefore, the plasma membrane is less fluid. Third is degree of unsaturation. That means whether the fatty acid chain is saturated or not. When the fatty acid chain in the phospholipid has double bond, then it is known as unsaturated, and hence kink is present. So they are less tightly packed and their van der Waals force of interaction is also less. So the membrane is more fluid. And if the fatty acid chain is saturated, that means no double bond is present, no kink, hence they are more tightly packed, so more van der Waals force of interaction, therefore membrane is less fluid. Cholesterol is present in the plasma membrane of animal cells and it affects the fluidity of the membrane. Cholesterol, it is present in plasma membrane of animal cells. This is phospholipid molecule and these are cholesterols. When the temperature increases, we know that membrane becomes more fluid because there is the more movement of the phospholipid molecules. Presence of the cholesterol interferes with this movement and hence it decreases its fluidity. When the temperature decreases, we know that membrane becomes less fluid. Presence of the cholesterol interferes with the interaction between fatty acid chain. So, cholesterol prevents membrane from freezing, so it increases fluidity of membrane at lower temperature in the presence of cholesterol. Cell coat or glycocalyx, carbohydrate coating that is present on the surface of eukaryotic cell is known as cell coat or glycocalyx. This is eukaryotic cell, nucleus, cytoplasm. Inside there is plasma membrane and outside there is cell coat or glycocalyx. This cell coat or glycocalyx is made up of carbohydrate that is present on the outer surface of plasma membrane in the form of glycoprotein, glycolipids, and proteoglycans. It protects the cell from chemical and mechanical damage. And also helps in specific, transient, cell adhesion. We are going to discuss about protein component of plasma membrane. There are two types of membrane protein, peripheral protein, also known as extrinsic protein, and integral protein known as intrinsic protein. Peripheral protein can be removed from the phospholipid bilayer by the gentle extraction procedure. Example: solutions of low or high ionic strength or of extreme pH. But these integral protein cannot be removed by these gentle extraction procedure. When we remove peripheral protein, lipid bilayer remains intact. But when integral protein is removed, lipid bilayer is disrupted. Peripheral protein do not interact with hydrophobic core of phospholipid bilayer, because these peripheral proteins are either attached to the polar head region of the phospholipid bilayer, or these are indirectly attached to the integral proteins. These interact with hydrophobic core of phospholipid bilayer. Here we can see that these two are integral proteins which interact with the hydrophobic region, because some of their portions are embedded in the phospholipid bilayer and hence they interact with the hydrophobic region of phospholipid bilayer. Peripheral proteins are soluble in aqueous solution. Integral proteins are insoluble in aqueous solution. Peripheral proteins are non-covalently attached to phospholipid bilayer, but integral proteins are covalently or non-covalently bound to phospholipid bilayer. Integral proteins are of two types: transmembrane protein and lipid link protein. Transmembrane protein spans entire phospholipid bilayer, and the force that holds this transmembrane protein is hydrophobic force. Lipid link protein, these are covalently attached to lipids. Transmembrane proteins are further of two types: monotopic transmembrane protein or single pass. These spans the phospholipid bilayer only once. So this is monotopic transmembrane protein. Second is polytopic transmembrane protein, also known as multipass. These spans the membrane more than once. This is polytopic transmembrane protein. It is spanning the membrane, or it crosses the membrane more than once. In case of the transmembrane protein, membrane spanning region of protein is mostly alpha helix, but in some cases it is beta barrel. Example in porins. Lipid link protein, these are covalently attached to certain class of lipids. First is isoprenoid group. Proteins are attached to either farcinyl residue, that is 15 carbon fatty acid, or geranyl residue, that is 20 carbon fatty acids, and these are attached by the cysteine residue present at the C terminus by thioether linkage, and the location is cytosolic, or we can say intracellular. Second, myristoyl or myristic acid. This is 14 carbon saturated fatty acid. Proteins are attached by the glycine residue present at the N terminus. Linkage is amide linkage, and their location is cytosolic or intracellular. Then palmitoyl or palmitic acid. This is 16 carbon saturated fatty acid. Proteins are attached by cysteine residue present near the N or C terminus by thioester linkage, and the location is cytosolic. GPI linked or glycosyl phosphatidyl inositol linked. Proteins are attached by various residues present at the C terminus, and the linkage is amide linkage. These are present at the cell surface. Proteins are classified on the basis of their function. One is transport protein. These spans the membrane and forms the hydrophilic channel and allows the selective transport of the solute across the membrane, or some of them hydrolyzes ATP and uses this energy for actively pumping the solute across the membrane. Some of the proteins present in the membrane also function as the enzymes and catalyzes certain metabolic processes. In some cases, more than one proteins are present that function as the enzyme, and then they catalyze different steps of a certain metabolic process sequentially. Protein as receptor sites. Some of the membrane protein has receptor sites that are present outside the cell, to which chemical messenger comes and binds. It can be hormone. If this membrane protein spans the membrane, then the binding of the chemical messenger results in the change in conformation of the region of the protein that is present in the cytoplasm, and this change in the conformation results in the activation of this region of protein. Then this initiates series of chemical reaction in the cell. Fourth is cell adhesion. Adjacent cells are adhered at the intercellular junction by the adherence of membrane protein that are present in the adjacent cells. This is the membrane protein of the first cell and this is the second cell. Fifth is attachment to cytoskeleton. Membrane proteins are bonded to actin filaments or other elements of cytoskeleton. This is membrane protein and it is bounded to cytoskeleton. This helps in maintaining the shape of the cell and also in some cases helps in fixing the location of certain membrane proteins. Let us see transport protein. These are responsible for selective transport of molecules. There are two types of transport protein. Carrier protein and second is channel protein. Transport through carrier protein is active or passive. Transport through channel protein is passive. Rate of transport is less. In case of channel protein, rate of transport is much faster. Transport through carrier protein is more specific. Transport through channel protein is less specific. In case of carrier protein, saturation is achieved. In case of channel protein, saturation is not achieved or saturation is less. Depending upon the number of solute molecules that are transported by carrier proteins, there are two types of the transport: uniport and co-transport. Uniport. In this, one solute molecule is transported from one side of the membrane to the other side of the membrane. This is uniport, and this molecule is transported from the one side of the membrane to the other side. Co-transport. When the transport of one solute molecule is coupled with transport of other solute molecule. It is known as co-transport. Depending upon the direction of movement of two solute molecules, co-transport is of two types: symport and antiport. When the transport of one solute molecule is coupled with the transport of other solute molecule in the same direction, it is known as symport. Antiport. When the transport of one solute molecule is coupled with the transport of the other solute molecule in the opposite direction, it is known as antiport. So, this is all for today's video. In the next part, we will discuss about membrane transport. So, please keep watching and also like, share and subscribe. Thank you.